Bismillahirrahmanirrahim. Hello to everyone. My name is Nourain. I am a student currently pursuing my MPhil in the microbiology department second semester with registration number MB320222020. I have the privilege of my presentation topic on gene expression in microbes to Dr. Niaz Muhammad, the esteemed chairman of MLT department. Let's begin with an introduction to gene expression in microbes. Gene expression refers to the process of which genetic information is used to produce functional molecule. Gene expression is a fundamental process in which the information stored in a gene is utilized to create functional molecule like protein or non-coding RNA. Imagine a gene has a library containing instruction like a blueprint for building specific molecule. In this mechanism, two main stages are involved: transcription and translation. Transcription: a segment of DNA known as gene is transcribed into a complementary RNA. This RNA acts as messenger or carrier the genetic instruction encoded in the gene. It serves as a template for next stage, translation. Translation occurs in the cell cytoplasm. The RNA molecule guides the synthesis of protein, which play a crucial role in performing a variety of functions essential for microbial survival and growth. Understanding of microbial genes are expressed how they are turned off and they are turned on. In the context of disease-causing bacteria, the mechanism that control gene expression can help us develop strategies and control these pathogens by manipulating gene expression. Now, using gene expression to control disease. Many strains of bacterium Staphylococcus have been resistant to penicillin, which poses a significant challenge in treating infections caused by these bacteria. One of the mechanisms that confer penicillin resistance in these bacteria is the production of bacterial protein called beta-lactamase. Beta-lactamase is an enzyme that has the ability to break down and deactivate penicillin. However, what interesting is that gene responsible for producing beta-lactamase is only expressed or only turned on when the presence of penicillin is happen. If there is penicillin, the gene will be turned on, and if bacteria are not exposed to penicillin, the gene will be turned off and no beta-lactamase will be produced. Genetic diversity in prokaryotes. Random mutation can occur during DNA replication period to cell division. This mutation introduces variation in the genetic code, resulting in different traits or characteristics in the daughter cell. This genetic diversity is a driving force behind the ability of prokaryotes to adapt and evolve in response to change in their environment. If environment change or if new selective pressure arises, those individuals with beneficial mutation have a higher chance of survival and passing on their genetic trait for future generation. Now, replication mistakes and mutation gene. Further, this genetic diversity plays significant role in the process of natural selection. Individuals with traits that confer advantages in their environment and more likely to survive and reproduce, leading the propagation of those advantageous traits within the population. Traditionally, mutation has been considered as a bad news because they can introduce error or disruption in the genetic code. Most mutations have determinantal effect on the organism function or survival. However, occasionally a mutation can result in protein that possess a property that improve the organism ability to survive and reproduce. An example of such mutation is found in the beta-lactamase gene of Staph bacteria. This gene mutation resulted in the production of enzyme called beta-lactamase, which can deactivate penicillin and confer resistance to the antibiotic. This advantageous mutation allowed providing them with a selective advantage over the other bacteria that lacked this mutation. I would like to discuss two important mechanisms that contribute to increasing genetic diversity in prokaryotes. Number one, vertical gene transfer and horizontal gene transfer. Vertical gene transfer is a way organisms pass on their genes to their offspring. It's like a traditional inheritance process observed in most organisms, including prokaryotes. During vertical gene transformation, genetic information is replicated and passed down from one generation to the next. This helps ensure the continuity and evolution of species. On the other hand, horizontal gene transformation involves the transfer of genetic material between organisms that are not direct descent. In the horizontal gene transfer contain three types: transformation, transduction, bacterial conjugation. Transformation occurs when bacterial cell takes up free-floating DNA from its environment. Transduction involves the transfer of genetic material through viruses called bacteriophage, and bacterial conjugation is a process where genetic material is transferred between two bacterial cells that are in direct contact with each other. These mechanisms of horizontal transformation play a significant role in prokaryotic ability to acquire new traits and adapt to changing environment by exchanging genetic material. Prokaryote can acquire beneficial gene or genetic variation that enhance their survival and evolutionary potential. Now, an experiment that is performed by Frederick Griff to check increasing genetic diversity in prokaryote. This experiment was conducted in 1928. Explaining that experiment aimed to investigate the nature of genetic material. Griff worked with two strains of Streptococcus pneumonia, a bacterium that causes pneumonia. Streptococcus is a bacteria that causes pneumonia, and they have two strands. Number one, virulent, which causes disease of pneumonia, and the other one, non-virulent, which they do not cause that disease of pneumonia. So what they do in step number one, Frederick inject the non-virulent strain to the mice, and as a result, mice alive. Frederick take the rough strain of Streptococcus pneumonia, so as a result, the mice alive. In the second step, Frederick inject the virulent strain, which the mice developed pneumonia and dead. In the third step, Frederick take the heat-killed smooth strain of Streptococcus pneumonia. Griff inject heat-killed virulent strain. So as a result, what they observe? They observe the mice alive. In the fourth step, Frederick inject the heat-killed virulent and rough strain of Streptococcus, which both do not ability to cause pneumonia disease. But what they observe? They observe the mice dead, because what happened? There is the genetic transformation between the heat killed and rough strain of bacteria strain. He stated that surprisingly the mice became sick and died. So conclusion of this whole experiment, there must be a substance in a heat kill bacteria that transferred the non-virulent bacteria. And Frederick mentioned that he called this substance the transferring principle, which carrying the genetic information responsible for the virulent strain. These are the whole experiment which is done by the Frederick. Thanks for your attention.